Welcome back friends, welcome back in another video tutorial from microbiology with Chagnik. So in this video tutorial, we will going to talk about a new concept of our protein, that is protein folding. So let's get started with the video friends. So first, let's start with the very basic thing, that is protein, or the very basic unit of protein is amino acid, right? Protein is made up of what? Protein is made up of amino acid. Now this amino acid will going to connect with each other via what? Via peptide bond, which is mainly a amide bond, right? And that's how a long peptide chain is formed, right? If that long peptide chain contains more than 10 amino acid, we will call it polypeptide chain, right? And this polypeptide chain is the primary structure of protein, right? Now this polypeptide chain can attain a secondary shape, that may be alpha helical, that may be beta sheet, that may be 310 helix, that may be pi helix, whatever. We have already made video on those things. You can also watch those video to understand. So this primary structure will attain this kind of things by the help of hydrogen bond, right? And this type of structure is known as secondary structure. After that, what will happen? After that, it will be converted into its three-dimensional tertiary structure, okay? And the conversion of the unfolded polypeptide chain into its folded three-dimensional tertiary structure is known as protein folding. Understood? So now, why we are studying protein folding? So if you remember our discussion on the native polyacrylamide gel electrophoresis, then I hope you may remember that in that particular native page, in the utility section, we have discussed that native page. In the native page, the biological activity of a protein can be determined, right? And if you remember that in that native page, in composition, we have not used any kind of denaturing agent, right? That means in native page, we are allowing the protein to run into its three-dimensional shape. Why is this so? Because remember it very carefully, for a particular protein to work, it is important that this particular protein should be in its native tertiary structure, okay? And this tertiary structure is actually achieved by this protein folding process, okay? And this protein folding is very important. If somehow the folding becomes wrong, then it will be very dangerous for that protein, not only for that protein but also for the organism, because this failure of folding will convert that particular protein into its inactive form, okay? And that may hamper the metabolic process in which that particular protein is taking part, right? So that is how much important a protein folding process is. So it is very important to understand for us, right? Because try to think about this, if we going to crack the how a protein folding thing can be arrested, then we can do a lot of things, right? For example, suppose a protein becomes cancerous, okay? So we can easily inactivate it with the help of the idea of how a protein can be unfolded, right? So that it cannot no longer work, right? That is the idea how much the protein folding is very useful. Although we will going to talk about very basic part of the protein folding. So protein folding is also depends on the nature of the solvent, the concentration of the salt, and obviously the presence of molecular chaperone. We will going to talk about this molecular chaperone things in our upcoming videos. So now let's come to the main topic of today's point, that is the Christian Anfinson's and his colleagues experiment, which is the first and most basic experiment to study protein folding. Okay, so what he have done, he have isolated RNase A, that is ribonuclease A, from bovine pancreas which is an enzyme of molecular mass 13,700 Dalton, and this particular protein consists of 124 amino acid with 4 disulfide linkage. So what he have done, we will going to learn it with the help of a diagram. So I have taken this diagram from CSIR Pathfinder book. You may also refer this book to understand the concept. So he have mainly studied how a protein refolds. What he have done to study the refolding of protein? First of all, he have taken the native ribonucleus. Okay, let me zoom the picture. Okay, that's good. So he have taken the native ribonucleus. Then he have also added 8 molar urea and beta mercaptoethanol. So 8 molar urea is a denaturing agent that will break all the weak interaction in between the polypeptide chains in that particular protein, and beta mercaptoethanol is a reducing agent that will break the disulfide bond, right? If you have studied the SDS page, then you may know about the beta mercaptoethanol. So as a result, what will happen, this native ribonucleus in presence of this urea and beta mercaptoethanol will be completely denatured, right? Now he have performed two sort of experiment. In the first sort, what he have done, he have removed both urea and beta mercaptoethanol, and as a result, what he have observed that this denatured ribonucleus is fully converted into native ribonucleus, meaning that, or this first sort of experiment concludes that this amino acid sequence should have all the necessary information for protein folding. That's why when urea and beta mercaptoethanol removed, this denatured ribonucleus is converted into its native form simultaneously and on its own, right? Not simultaneously, but it is spontaneously, right? Because it should take some time. Now in the second sort of experiment, what he have concluded, in the second sort of, first learn that one, what he have done in the second sort of experiment. So in the second sort of experiment, he have only removed the beta mercaptoethanol, but he have conducted the experiment in presence of 8 molar urea, and as a result, what he have find that the 4 disulfide linkage which was formed in case of native ribonucleus was also formed here, but the twist is here the disulfide bond is formed in between incorrect partners. Okay, that means the positioning of disulfide bond is not correct in that case. Okay, that simply concludes that the weak interaction which was broken down by 8 molar urea was very important for the correct positioning of disulfide bonds. Not only that, he was also concluded that when he have conducted the experiment in presence of 8 molar urea, he have observed that the structure that the denatured ribonucleus is attaining is quite different from the native ribonucleus. That means the weak interaction also have a very important role in assuming the native shape of the protein, right? So that is the first experiment of Christian and Finson that tell us a lot about what? That tell us a lot about the protein folding. Okay. So now in the second part of this video, we will going to talk about the molten globule model. So after the Christian and Finson's experiment, there is one question that is arising. The one question is, if the amino acid sequence have all the necessary information for folding, then why that particular unbranched polypeptide chain is attaining only a one shape, despite it can form into a number of endless conformation, right? Because it is just an unbranched polymer, right? It is just a small chain. It can fold into any way that it wants. Then why just only this one? That is the first question that was arising. And so there are various type of models that was proposed to explain how this folding is done. So most of the models failed, but there are very important models also available. that guide us to understand how this folding occurs. Among them, we will going to talk about the very basic model, that is the molten globule model. So what this model tells us, this model tells us, we will going to understand with the help of a diagram, which is again I have taken from that CSI 100 Pathfinder book. This model tells us that the unfolded protein will be converted into molten globule state. This is a state which is not similar to the denatured state of the protein, and also it is not similar to the native state of the protein. It is some kind of a intermediate state. Okay, so and the conversion of molten globule state from the unfolded state is very fast. It occurs within a few milliseconds. Okay, that is why what is happening in between unfolded and molten globule is not still known. Okay, but we have already understand that some basic characteristics of molten globule that is quite very important and interesting. That is, this molten globule state have all the secondary structure that is present in native state. Okay, and importantly, this secondary structure is not that much compact as it is in case of native state. And the most importantly, as you can see in this picture, the packing interaction in the interior of the molten globule is not that much compact and good as in case of the folded state. Okay, so that is the difference between the molten globule state and the folded state. And after that, from that molten globule state via a slow step, it will be converted into the folded model. Okay, so this is the very basic model how we can talk about the protein folding. So this is the concept of our molten globule state. So if you see this thing, you can also, it will be very clear to you. That is, the molten globule state has the most secondary structure of the native state, but it is less compact and the proper packing interaction of the interior of the protein have not been formed. And this event is very fast. That is why we know nothing about how it is going on, but we know some characteristics of the molten globule. For example, secondary structure is almost present as it is in case of native state, but it is less compact than native structure. And most importantly, the proper packing interaction is not going on in case of molten globule state, right? The interior side chain in case of molten globule state is mobile and more closely resembling the liquid as compared to the solid like interior of the native state. So that is the thing. So you can just remember that unfold state with the help of a very fast reaction converted into molten globule state, which is a intermediate step. In that step, it have all the secondary structure that a native state contains, but it is not that much compact and the packing interaction in the interior have not been formed, right? And then from that molten globule state via a slow step, it will be converted into fully folded state. Now that particular protein is fully active and it can function biologically, right? So that is the idea of our protein folding. So I hope this video will be helpful for you. Thank you.